Hi, my name is Lauren Svejcar. I work for Oregon State University at the Eastern Oregon Agricultural Research Center, and today I'll be talking about the use of priming with novel seed enhancement technologies in the Great Basin. Restoration efforts using native plant materials in degraded western rangelands have been extensive, yet success rates are chronically low due to two primary constraints. One, fall plantings may result in seed germination and subsequent mortality from winter freezes. Second, spring plantings may result in late germination and insufficient time to establish before the high temperatures and drought conditions of summer. So how do we overcome these limitations? Seed enhancement technologies that produce extruded seed pellets or pillows using various absorbent filler materials, biostimulants, plant protectants, and binders are being developed to overcome these constraints by enhancing the seed and seedling microenvironment. Additionally, seed priming techniques which allow seed germination processes to begin by partially hydrating seed at a water potential that allows imbibition but prevents radical extension may provide an advantage for native seedling establishment. Seed enhancement technologies such as extruded pillows and pellets are comprised of dry and liquid materials. Dry materials often include calcium bentonite, diatomaceous earth, compost, worm castings, and superabsorbent powders. Similar to baking, the dry materials are thoroughly mixed together. Then seed and liquids, including water, cell-vol, and fungicides, are added to the dry ingredients and mixed to form a dough. Extruded pellets and pillows are made using industrial machinery, including dough extruders, to the left, and pasta machines, image to the right. Pillows and pellets help to create conduits for seedling root growth in soils that form hard physical crusts, increase water infiltration potential around the seedlings, and provide nutrients for seedling growth. The objectives of our research are to test the utility of priming techniques with seed enhancement technologies to overcome environmental limitations. Specifically, we were looking to develop and test priming techniques combined with seed enhancement technology materials on native grasses, forbs, and shrubs. And second, to compare seedling emergence between extruded seed pillows with primed and non-primed seeds and non-treated seed in a controlled laboratory setting. Solid matrix priming techniques were used for native plant species using seed enhancement technology materials that form the pellets or pillows. A moisture release curve was developed for the material using a WP4C dew point potentiometer from Decagon devices. Data from the moisture release curve was used to test for optimal conditions, or optimal water potential, at which to prime various cool season bunchgrass species and growth chambers. We set our growth chambers at 15 degrees Celsius with 12 hours of daylight. Once optimal conditions were determined, primed seeds were processed according to seed enhancement technology standards for laboratory validation. Lab studies were conducted in an environmental grow room set at a constant temperature of 21 degrees Celsius with 12 hours of daylight simulated using fluorescent lighting. Field soils from 3 different geographic locations on the Kaibab Plateau in northern Arizona were put into 16-liter wooden boxes with 4 seed treatments, including a control, planted at 3 different depths using a randomized block design. 2 laboratory studies were conducted in the environmental grow room. The first study was an initial analysis of priming techniques using a short-duration trial of Poa fundleriana and Pseudorogenia spectata. Soils were sandy loam, one each with low and high organic matter. In a second study, priming techniques were compared to drill-seeded treatments with plantings of Elymus elamoides and Elymus trachycaulis at 3 depths, 0, 10, and 30 millimeters. Soil from 3 landscape positions was used: ridgetop, hillslope, and valley bottom. Watering regime was 1 centimeter every 10 days. In the first lab study, Poa fundleriana and Pseudorogenia spectata, seeds that were either left untreated, control, incorporated into a pillow or primed and incorporated into a pillow, were measured repeatedly for plant density. Primed pillows had faster emergence overall and produced higher plant densities than the control. Above-ground biomass measurements were collected for Elymus elamoides and Elymus trachycaulis from treatments of control seed, untreated, coated seed, agglomerated seed, and seed primed and incorporated into a pellet. Treatments were sown at 3 different planting depths in 3 different soils. Plant growth was typically higher for prime pellets at all sowing depths and soil types. Ongoing research includes field projects that have been implemented in Oregon, Nevada, and northern Arizona to evaluate the utility of prime pellets and pillows for overcoming environmental barriers to seeding success. Additionally, laboratory and field studies evaluating the potential of priming seeds in herbicide protection pods and altering pillow and pellet shape for increased water absorption potential are currently underway. Based on preliminary studies, we believe there is potential for seed enhancement technologies to yield direct and significant financial savings to land managers by improving seedling establishment rates and reducing the time and amount of seed required for seeding projects. Use of priming in the development of seed enhancement technologies is currently proving to be a successful strategy for overcoming primary limiting environmental factors. Continued research of this technology will further improve advances and success of rangeland reseeding efforts in the United States and abroad. We are very appreciative of the work and effort provided by Eastern Oregon Agricultural Research Center scientists and staff, with funding provided by the U.S. Department of Agriculture, the Nature Conservancy, the Grand Canyon Trust, and Aquatrolls Corporation. Thank you for your time.